Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, imagine this. You need to find one specific sentence in a massive library. Before 1985, finding a tiny bit of DNA was kind of like that, but way harder. A huge, multi-day job, needing lots of material, sometimes even radioactive stuff.
Yeah, it was seriously laborious.
Then, bam, this paper comes out. And it introduces something totally new, almost like a DNA photocopier.
Exactly. That's the perfect analogy.
And suddenly, you could take just a tiny trace of DNA, stick it in a test tube, and make millions, even billions of copies of just the bit you wanted. In hours, not days.
It changed everything. I mean, think about genetic tests, forensics, even how we tackled COVID-19 testing. It all comes back to this core idea.
Welcome to the deep dive. We unpack complex stuff and pull out the key insights for it.
And today we are diving deep into that foundational 1985 science paper, the one titled Enzymatic Amplification of B globin Genomic Sequences and Restriction Site Analysis for Diagnosis of Sickle Cell Anemia.
Right. And we've also got an innovation analysis report to help us. Our mission today, to really get our heads around the sheer brilliance behind PCR, the polymerase chain reaction, and figure out why it's seen as, well, one of the biggest breakthroughs in biology ever.
And, whether you're just curious about how a genetic test works or you love a good story about scientific light bulb moments, this is for you. We want to give you a really clear picture of a technique that just completely reshaped how science gets done.
It really did. Okay, so to appreciate just how massive PCR was, let's rewind. What was life actually like for molecular biologists before 1985? Especially if they were trying to find something like the sickle cell mutation, just one single letter change in the DNA code, right? An A swapped for a T.
Oh, it was. Painstaking. Honestly, frustrating is probably the better word. You were looking at techniques like southern blotting, which is complex, or really, really time-consuming DNA cloning.
And these methods, they needed a lot of starting material.
A ton. We're talking micrograms of DNA. That often meant pretty large blood samples from patients, not just a quick cheek swab like today.
Wow, micrograms. So what did that actually mean, practically speaking, for the patient, for the doctor waiting for results?
It meant, well, first, a significant blood draw, and then a long wait. The lab work itself took days, sometimes even weeks. Plus, many of these techniques, like southern blotting, used radioactive labels.
That's right. I remember hearing about that.
Yeah, so you had safety concerns for the lab staff, handling radioactive materials, developing films in dark rooms. It all added up. Slow turnaround times were a huge bottleneck for getting diagnoses or making clinical decisions.
Okay, so the need was clearly there. But scientists kind of knew about the building blocks, didn't they? They knew about enzymes, DNA polymerases that could copy DNA strands. They knew about primers. Why hadn't anyone put it together before? It seems almost obvious now.
Hindsight is always 20/20, isn't it? The pieces were there, sort of. But the huge hurdle, the real knowledge gap, was figuring out how to make it specific and exponential. How do you amplify only the tiny segment you care about, millions of times, starting with maybe just a few copies mixed in with the entire genome, and do it easily in one tube?
So it's the specificity and the exponential growth from tiny amounts.
Exactly. People knew polymerases could copy DNA, but controlling it, targeting it precisely, and making it cycle over and over to get that massive amplification without amplifying everything else. That was the puzzle nobody had solved. Plus, the enzymes they typically used back then weren't stable at high temperatures, which, as we'll see, was crucial.
And there was real pressure to solve it, right?
Yeah.
A clinical need for tests that were faster, safer, needed less sample.
Absolutely. Transforming genetic testing, enabling things like prenatal screening for inherited diseases, identifying carriers, even forensic applications. The potential was enormous if someone could crack this enzymatic amplification problem. It was about ditching those cumbersome, slow, radioactive methods for something elegant and fast.
Okay, so let's get into the breakthrough itself. The core idea of PCR. This is where Kary Mullis comes in, right? The chemist with the famous late-night drive inspiration.
That's the story. Whether it happened exactly like that or not, the conceptual leap was his. The idea that you could use repeated cycles of primer binding and DNA synthesis to exponentially amplify a specific DNA target between those primers. It was quite unconventional thinking at the time.
So what did that look like in practice, the design?
Okay, so the key was using two short, custom-made DNA sequences called oligonucleotide primers. Think of them as like starting flags or GPS coordinates. One primer matches a sequence on one strand of the DNA target, and the other primer matches a sequence on the opposite strand, a short distance away. They essentially flank the region you want to copy.
Got it. So they bracket the target sequence, then comes the cycling part, the photocopying process.
Yeah.
How does that work, step by step?
It's basically a three-step loop, repeated many times. Step 1, denaturation. You heat the DNA sample, maybe to around 95 degrees Celsius. This heat breaks the bonds holding the two DNA strands together so they separate, like unzipping a zipper.
Okay, strands separated. What's next?
Step 2, annealing. You cool the mixture down, maybe to somewhere between 50 and 65 degrees Celsius. This lower temperature allows those short primers, which are present in huge excess, to find and bind or anneal to their specific matching sequences on the now single DNA strands.
So the primers stick to their targets,
Yeah.
and then?
Step 3, extension or elongation. You raise the temperature slightly, maybe to 72 degrees Celsius, which is the optimal temperature for the DNA polymerase enzyme. This enzyme latches onto the primer, which acts as a starting point, and begins adding DNA building blocks, synthesizing a new strand complementary to the template strand. It copies the DNA stretch between the two primers.
Okay, denature, anneal, extend. And the magic happens when you repeat this cycle.
Precisely. That's the exponential part. After the first cycle, you've doubled the number of target DNA molecules. But now, those newly synthesized strands also serve as templates in the next cycle.
Ah, I see.
So in cycle 2, your original templates get copied again, and the copies you made in cycle 1 get copied. So you go from 2 copies to 4, then 4 to 8 in the next cycle, then 16, 32, and so on. It's 2 to the power of n, where n is the number of cycles.
Wow, so after, say, 30 cycles, you'd have over 1 billion copies.
Theoretically, yes. You get this incredible amplification from potentially just a single starting molecule. That's the power. Going from nearly undetectable to easily detectable quantities in just a couple of hours.
Mind-blowing. Okay, so they had this amazing concept. How did they prove it worked in this 1985 paper? They focused on sickle cell anemia, right?
Yes, that was the perfect test case. A well-understood genetic disease caused by that single A to T change in the beta-globin gene. First, they had to show the amplification was actually happening and that it was exponential, or close to it. They did experiments measuring the DNA yield after different numbers of cycles and showed it increased dramatically, consistent with the theory.
So they proved the photocopying worked. Then how did they use it to diagnose sickle cell?
They designed their primers to amplify a specific 110 base pair fragment of the beta-globin gene, right where that mutation occurs. Then, here's the clever bit. That single A to T mutation either creates or destroys a cutting site for a particular enzyme, a restriction enzyme called mTII.
Okay.
So they took the amplified DNA, the billions of copies of that 110 base pair fragment, and treated it with the mTII enzyme. If the person had the normal gene sequence, the enzyme would cut the fragment into 2 smaller pieces. If they had the sickle cell mutation, the enzyme couldn't cut. If they were a carrier, heterozygous, they'd get a mix of cut and uncut fragments.
Ah. So you could just run the results on a gel and see the pattern of DNA bands, cut, uncut, or both.
Exactly. It gave a clear, direct visual readout of the person's genotype, normal, carrier, or affected, from just a tiny starting amount of DNA in a matter of hours. No more multi-day southern blots, no radioactivity.
That sounds like, well, a revolution. Let's list the advantages again.
Okay, number 1, DNA input. Reduced by about 1,000 times, micrograms down to nanograms.
Huge.
Number 2, speed. Days or weeks shrunk down to a few hours.
Game changer for diagnostics.
Number 3, safety and simplicity. No need for radioactive probes. It was an enzymatic reaction in a tube.
And number 4.
It wasn't just making an old method better. It was a totally new concept. In-tube enzymatic signal amplification. A whole new way of thinking about detecting specific sequences.
Incredible. Now, this didn't happen in a vacuum. You mentioned Cetus Corporation. Tell us about the team and the environment.
Right. Cetus was key. It was an early biotech company, and they had resources like automated machines for making the oligonucleotide primers and early versions of thermal cyclers machines that could automatically change the temperatures for the PCR cycles. This industrial setting was critical, but they also collaborated with academics, particularly from UC San Francisco.
So industry muscle plus academic brain power. Who were the key people?
Well, Carey Mullis, obviously the conceptualizer, the one with the, let's say, unconventional spark. But you also absolutely needed Randall Saiki. He was the meticulous experimentalist, the guy at the bench doing the careful primer design, running the gels, making sure the results were solid and reproducible.
The practical hands.
Exactly. And then there was Henry Ehrlich. He was the bridge to the clinic. He saw the diagnostic potential, helped frame the application for sickle cell, importantly, secured the patient DNA samples needed to actually test it on real-world material, and understood the medical relevance.
So it was that combination, the creative idea, the rigorous bench work, and the clinical application focus.
Precisely. That synergy within that sort of industry-academia microcluster allowed them to go from idea to published proof of concept, demonstrating clinical utility incredibly quickly, way faster than might have happened in a purely academic or purely industrial setting, perhaps. Having those patient samples readily available was also a huge accelerator.
So this paper really did mark a paradigm shift, as you said, not just improving something, but introducing a whole new way of doing things, the DNA photocopying principle that could, in theory, be applied to any DNA sequence.
Absolutely. Now, it wasn't perfect right out of the gate. That first enzyme, the Clinau fragment, wasn't heat stable. Remember, the denaturation step is hot, around 95 degrees.
Right, so the enzyme would die each cycle.
Pretty much. They had to manually add fresh enzyme after the denaturation step in every single cycle. Imagine doing that 30 times.
Oh, wow. Tedious and prone to error, I bet.
And risk of contamination. But that problem was solved fairly quickly, within about 5 years. The really big breakthrough post-Mullis was the use of a heat-stable DNA polymerase, Taq polymerase, isolated from a bacterium found in hot springs, Thermus aquaticum.
Ah, Taq. I've heard of that.
Taq polymerase could withstand the high denaturation temperatures. This meant you could put everything in the tube at the start, DNA, primers, building blocks, and Taq enzymes seal it, put it in an automated thermal cycler machine, and just let it run. That revolutionized PCR, made it easily automatable and much more robust.
And that automation really blew the doors open for applications.
Totally. It paved the way for everything we associate with PCR today. Cloning specific genes became way easier. Genetic testing and screening prenatal, carrier status became routine. Forensic DNA fingerprinting took off.
Right, the CSI effect.
Kind of. And it went beyond that. Studying complex microbial communities in the environment metagenomics became possible without needing to grow the microbes in the lab. And, as we all experienced, rapid diagnostics for infectious diseases like HIV, influenza, and most famously, COVID-19, PCR became a household name.
It really did. The impact is just undeniable, which of course led to the ultimate recognition.
Yep. The Nobel Prize in Chemistry for Carey Mullis in 1993, acknowledging just how profoundly PCR had changed molecular biology and medicine.
So wrapping this up, what we've looked at today isn't just, another scientific paper. It's the origin story of a technology that fundamentally altered our relationship with DNA. From that spark of an idea, maybe on a night drive, maybe not, to a Nobel Prize-winning technique that's now indispensable. PCR took genetic analysis from this slow, clunky, specialist process and made it fast, accessible, and essential.
It really did. And the core beauty of it is that combination of, well, elegant simplicity in the concept, just cycling temperatures, and letting an enzyme do the work, coupled with that incredible exponential power. It makes you wonder, doesn't it?
Wonder what?
Well, if this relatively simple, iterative, enzymatic process could amplify a genetic signal so dramatically, what other biological signals are we currently struggling to detect? Maybe low abundance proteins or specific cellular events? Could similar amplification strategies, maybe not identical to PCR, but inspired by it, transform those fields too, turning slow, difficult assays into something quick and routine?
That's a great question. It's that idea of looking at an old problem, detecting something rare with completely fresh eyes, focusing on amplification. A really powerful reminder.
Definitely something to think about.
Thank you for joining us on this deep dive. We hope this gave you a much clearer picture of PCR, truly one of modern biology's most important innovations.